Good morning or good afternoon, whatever your location, and welcome to the sixth session of the ARCS Forward and the National Scholar Speaker Series. Today, our topic is neurological diseases, and we'll be hearing from Amanda McQuade from the Orange County ARCS chapter and from Chantel Charlebois from the Utah chapter. I'm Dr. Catherine Plange, and I'm a neurologist. When I retired from the Neurological Institute in Phoenix 20 years ago, I wondered what would I do with my time. Fortunately, I had good friends who introduced me to ARCS, and I've been a member for 20 years. In case you don't know, ARCS stands for Achievement Rewards for College Scientists, and our mission is to advance science in the United States by giving financial awards to academically outstanding students who are studying science, technology, engineering, and medical research. In a few minutes, you'll know what I mean by academically outstanding when you hear from Amanda and Chantel. If you want more information about ARCS, visit our website at arcsfoundation.org. The ARCS Forward and the National Scholar Speaker Series is a new initiative of ARCS, started about a year ago. And it's our way of getting together scholars from chapters across the country who might be studying the same fields, let them share information, and then also share it with us. Our previous sessions have included prostate disease, the environment, and public health, and have been very informative. On behalf of the ARCS Forward Committee, thank you for being here and joining us today. As you hear the scientists speak, if you have questions, please put it into your chat box and we'll try to get to them at the end of the session. So let's get started, and first we'll hear from Chantel from ARCS Utah. Thank you, Chantel.
Thank you. All right, I'll share my screen. How's that look?
Perfect.
Awesome. Yes, hi everyone. I'm Chantel, and I'm a PhD student in the Biomedical Engineering program at the University of Utah. So the field of biomedical engineering covers a pretty broad topic, but in general, when you use engineering principles to improve medical diagnostics or therapies. Today, I'm going to talk about my graduate research, which is in a subfield of biomedical engineering called neural engineering. And neural engineering helps to better understand, repair, replace, or enhance neural systems. So specifically, I'm going to talk about how I use medical imaging, computational models, and patient data to better understand responsive neurostimulation for temporal lobe epilepsy. So epilepsy is a disorder that's defined by having two unprovoked seizures separated by 24 hours. And a seizure is this transient occurrence of abnormal, excessive, and synchronous electrical activity in the neurons in your brain. So it's a very disabling disorder that can be defined by having these persistent seizures. There can be adverse effects from the drugs used to try to stop seizures. It may come with cognitive problems, comorbidities like anxiety or depression, a restricted quality of life, and there's the risk of sudden unexplained death in epilepsy. It's a pretty common disorder with about 1 out of 26 people in the US developing this over their lifetime. And so when most people think of epilepsy, they think of a tonic-clonic seizure, which you may have seen where a patient ends up jerking and their whole body is jerking. So this is an example of a generalized epilepsy, which you can see here in the middle, that involves both sides of the brain. So you're going to have that abnormal electrical activity across the whole brain. But epilepsy is actually a spectrum and includes both generalized seizures that involve both sides of the brain, and then seizures that may start in a specific region of the brain, like in this image here, where we can see this red abnormal activity just in one specific region of the brain. So Seizures differ on a patient basis, their location, where they are in the brain, how they manifest clinically, the size of the seizure, and when they occur. And the goal of treating epilepsy is to stop seizures. So usually the first line of treatment are to try some anti-seizure medications. And these medications successfully treat epilepsy in about 70% of patients. However, this third of patients that don't respond to drugs are called have drug-resistant epilepsy. And so they've usually tried at least 2 or more anti-seizure drugs, and these drugs were not successful in stopping their seizures. So another type of treatment for epilepsy is resection surgery. And this form of treatment can be used for focal epilepsy. So those are those epilepsies where the seizures are located in one specific part of the brain. So here on the right, it's an example of a resection surgery. So we see this area of the brain in green where the seizures are starting, and this part of the brain will actually be removed. So again, this is a pretty successful therapy for patients who have the surgery. About 50 to 90% of them become completely seizure-free. But up to 50% of patients aren't candidates to have this type of surgery because removal of the tissue where their seizures are starting could lead to permanent and debilitating neurological deficits. So there's a need for alternative therapies other than anti-seizure medications and resection surgeries for patients to achieve seizure freedom, especially for those patients who are not suitable to have surgery. So this brings us to the responsive neurostimulation system. This is an FDA-approved therapy for focal epilepsy, so those where seizures are in one region of the brain. And you'll actually have electrodes placed inside of the brain that are connected to this battery implanted in the skull. And so this is a closed loop system. So from those electrodes that are implanted in the brain near where the seizure starts, it's always recording that electrical activity. It can then detect a seizure, and it will use electricity to stimulate that region of the brain to try to stop the seizure. And so it's responding in real time to try to stop those seizures from occurring. So this is the device and the patients implanted with this device that I'll be talking about for my research today. And so the patient's neurologist will actually be able to program the device to identify these specific patterns of electrical activity that identify seizures for that patient, and then program the electrical stimulation that they're going to use to try to stop those seizures. So about 3/4 of the patients who use this responsive neurostimulation system have a 50% reduction in the number of seizures they were having before they were implanted with the device. But about 1/4 of the patients still don't respond. And the whole goal of any epilepsy therapy is to be completely seizure-free. So it's great that most of the patients are having a reduction in the number of seizures, but again, our ultimate goal is to have them have 0 seizures or be completely seizure-free. And so the goal of my research is to understand why some of these patients are responding to the responsive neurostimulation system and others are not. And the first hypothesis that we explored was, is there a specific location in the brain that we should be stimulating that is associated with better seizure reduction than other locations in the brain? So we're going to look at this from 2 different viewpoints. We're going to examine the local area of stimulation and then the brain networks that are actually connected to those regions. So the patient population I'm going to be talking about is patients with temporal lobe epilepsy, and this is a subtype of epilepsy where the seizures are starting in a specific region of the brain called the temporal lobe, and I'm showing that here in red. It's the most common type of drug-resistant epilepsy, and this region of the brain is responsible for processing auditory information, memory, language, speech, and emotion. And I'll just point out some of these structures inside of the temporal lobe that you'll see in some of my slides. So here in this dark blue circle is called the amygdala, And this lighter blue region is called the hippocampus, and those are in the middle of the temporal lobe. So like I said, I'll be talking about using medical imaging in my research. And so how we use medical imaging is before the patient is implanted with this device, we get a preoperative MRI, and this lets us know the anatomy of that patient's brain. And then after they've been implanted with the device, we'll take a postoperative CT scan, and this will show where the electrode is in the brain. So then we can use some computational methods to align the preoperative MRI and the postoperative CT. And that way we know where the electrode is in relation to the anatomy. So you can see that here we have the patient's anatomy, and then we can actually see where the electrode is in relation to the anatomy in the brain. And then our next step is we want to know the region surrounding the electrodes that gets excited when we stimulate with electricity. And so we estimate this, and we call it the volume of tissue activated, or the VTA. And so that's these yellow circles surrounding the electrodes. So we do that for all of the patients. Most of them have 2 electrodes in the brain. So there's 4 VTA on each of the electrodes. Then our next step is we want to compare across all of the patients in our cohort. So we'll combine all of those VTA for the different patients in one space, so we can compare the locations across all patients. And then we want to know, are there any regions that are associated with better seizure response or worse seizure response? And so we'll do some statistical analysis to try to identify this sweet spot or hot spot where we may want to stimulate in the future. So here's some results, and these figures were actually looking at slices of an MRI. So you can see here in the corner, we have the amygdala, which I showed before in dark blue, is in pink, and the hippocampus in gray. And so these lines are showing slices through the brain at those regions. So again, you can see the amygdala outline in pink, and we're just looking at how many patients were stimulated in each of those regions of the brain. So we want to see what's the distribution of stimulation in the temporal lobe across all the different patients who have this device. And so we can see that the maximum number of overlaps, so where most patients are being stimulated, is shown in these bright yellow regions in the middle of the hippocampus. Next, we want to know, in these regions, are there specific areas that are associated with having a better seizure reduction than if you're stimulated there versus if you're not stimulated there? And so that's what we're looking at in this figure. Again, these are slices through the amygdala and hippocampus on an MRI, and you can see the amygdala in pink and hippocampus outlined in gray. And so if you see these red blobs or red clusters, that means if stimulated there, patients had better seizure reduction than patients who were not stimulated there. And the opposite is true for blue. So we can see some clusters of red areas, but it was actually very dispersed throughout, and there wasn't a really obvious statistically significant hotspot where we were able to say, you should stimulate here if you want to have better seizure reduction. So we built a model to see if we could predict the patient's seizure outcome based on where they were stimulated, based on that blue and red clustered map. But we were not able to predict seizure reduction based on the stimulation location. So getting back to our objective, where we're trying to understand why some patients respond to RNS and others don't, we were looking at the local area of stimulation, and we found that it was not predictive of seizure reduction. So that brings us to our second idea, that maybe it's not this local area of stimulation, but it's maybe the brain networks that are connected to that region. So I'll talk about that next. And here we'll use another type of medical imaging called diffusion weighted MRI. And so this actually allows you to identify these major neuronal tracts in the brain. So you can see which regions are connected to other regions by creating these tracks that represent connections in the brain. And so we did this for all of our patients. We used the diffusion weighted imaging, and we actually calculated the connectivity of the VTA, so that volume of tissue activated, to the rest of the brain. So regions close to the VTA, shown here in purple, have a high connectivity to the brain, meaning that there's a lot of those neurons that are connected to that region and the VTA, whereas more distant regions have a lower connectivity value. So then we wanted to understand if a region has high connectivity, is that associated with good response or bad response? And so we did a correlation of everyone's seizure outcome and their connectivity value. And that's what we're showing here on the right. So regions in orange mean if you have high connectivity to that region, you also have improved seizure reduction. So those are regions we would think we would want to be highly connected to. Whereas blue regions, if you're more connected to those regions, you have worse seizure outcomes. So we looked at this across our cohort, and we were able to identify these regions where we think strong connectivity was associated with seizure reduction. So those are these areas shown in orange, and blue are the regions that we would want to be less connected to. So again, we wanted to see if we could predict seizure reduction by overlaying a patient's connectivity map to this blue and orange map that we created. And we found that we were actually able to have a significant prediction of a patient's seizure reduction based on their connectivity map. So that was very exciting. Oops. And so then, now that we can say we could predict their seizure outcome, we wanted to know, well, what are these regions that are important in the response? What areas do we want to be connected to with stimulation? And so that's what this image is looking at. So these regions in red in the prefrontal cortex here are showing those regions that were associated with better response, whereas the blue regions are showing those with worse response. So we were able to identify specific brain regions that we are postulating may be involved in the response to seizure reduction for this type of therapy. So again, our hypothesis was to see if stimulation location was associated with seizure reduction. And we found that stimulation of specific brain networks was predictive of seizure reduction. So in conclusion, in this study, we looked at the local region of stimulation, and we found it was correlated with seizure outcomes, but not predicted. And this actually goes along with previous work that has been done by other groups, finding no correlation with that electrode location relative to that gray structure, the hippocampus. Then we looked at the network effect, and we found that patient-specific stimulation of specific networks was associated with seizure outcomes. And then we further were able to identify these regions that we call the prefrontal cortex, cingulate, and precuneus that may increase seizure reduction if there's high connectivity to these regions. So we're hypothesizing that maybe in the future we could run a study where we actually target these regions with stimulation and hope that patients would have a better response than they're currently having. So I'd like to thank the ARCS Utah chapter for funding me and my graduate research. It's been great getting to know the chapter members here in Salt Lake, as well as my other funding sources, my collaborators here at the University of Utah, and my mentors. Thank you.
Thank you, Chantel. You know, it looks like neuromodulation is a wave of the future. So it's very interesting to see you connecting it with resistant seizures. So thank you so much. Next, we'll hear from Amanda from the Orange County ARCS chapter.
Hello, everyone. I'm Amanda from ARCS OC, and I'm really excited to talk to you guys today about the work that I've been doing on Alzheimer's disease and actually the immune system as well. Can everyone see my screen?
Yes.
Perfect. So Alzheimer's disease is the most common form of progressive dementia, and it's currently the 6th leading cause of death in the United States and predicted to affect 75 million people by the year 2030. So this is a really, really important disease to be targeting right now. how Alzheimer's disease looks in the actual brain is a buildup of several types of proteins. So there's amyloid beta protein plaques, tau tangles within the actual neurons, and a severe loss of neurons and synapses in the brain. And so you can see in these little brains here that the Alzheimer's disease brain can end up shrinking to 2/3 of its original size. There's that many neurons that are lost. There's two main types of Alzheimer's disease. The first is familial Alzheimer's disease, and these people have an earlier age of onset. It's caused by a number of known genes, APP, PS1, and PS2. And these families have been really, really helpful in our ability to study how Alzheimer's disease begins, because we know that these people are eventually going to get Alzheimer's disease, but they only actually account for about 1% of 80 cases. Now, sporadic Alzheimer's disease is the vast majority of people. This is a later onset Alzheimer's disease. It's polygenic, so there's a lot of different genes that seem to influence it. And also there are some environmental causes as well. And it's very, very difficult to predict. So currently the treatments for Alzheimer's disease are not very good. There's a couple that can delay treatment or delay symptoms for a couple of years, but eventually this disease will take over again. And so there's really this desperate need right now to figure out something that can either stop the progression of disease from occurring in the first place, or try and reverse some of these changes later on. And the reason why it's so difficult to find therapies for Alzheimer's disease is because it's an extremely complicated disease. So we have these various different pathologies that are occurring in the brain, and they'll start to build up at different times. And by the time that someone actually knows that they have Alzheimer's disease, they're seeing this mild cognitive impairment. These pathologies have been building up in their brains for about a decade. And so it can be really, really difficult at the time that someone knows they have Alzheimer's disease to go back and try and reverse these changes that have been happening for the last 10 to 15 years. And so because of this, my goal is to study these very early onset parts of Alzheimer's disease. And I've done so by focusing on genetic risk factors. So we find genetic risk factors by studying single nucleotide polymorphisms. And so this is what's used for figuring out mutations in the DNA that may make it more or less likely that someone gets Alzheimer's disease. And so here's a plot of what this looks like for all the genes associated with Alzheimer's disease, where we have all the chromosomes down here lined up, and these different gene names that end up being associated with either protection or risk for Alzheimer's disease. And so when these studies were done a couple of years ago, it actually was really surprising to the field. People had thought that these Alzheimer's disease risk genes would all be expressed in neurons, since those are the cells that we use for thinking and the cells that are dying in Alzheimer's disease. But surprisingly, I'm circling here in red all of the Alzheimer's risk genes that are actually most highly expressed in cells of the immune system. So these main immune system cells in the brain are called microglia. And it seems like if you're changing expression of these microglia specific genes, that's actually what is changing people's risk for this sporadic Alzheimer's disease. So now the goal is to figure out how is the immune system actually impacting Alzheimer's disease? And microglia, these immune cells in the brain, have lots of different functions. They're involved in maintaining the health of neurons. They're involved in clearing dead cells and debris. They have different immune responses. And so we really need to understand which one of these immune functions or several immune functions are actually altered by these specific genes associated with disease, so that we can actually functionally come in and try and rescue what's happening here. And so at this point, we don't even know if you want more immune activation to help people with Alzheimer's disease, or if you want to stop this immune activation that is known to cure or occur with disease. And so that's sort of where I'm coming at this to study. Today, I'm going to be talking about one of these specific genes, which is called TREM2. The longer name is triggering receptor expressed on myeloid cells 2. They always have these horrible names for genes. And so I just want to talk a little bit about this gene. It's actually expressed on the surface of these immune cells, which will make it a little bit easier to target with therapies, and can actually, mutations in this gene increase your risk of Alzheimer's disease twofold. So for a gene that's already so prevalent, or a disease that's already so prevalent, this can be a really devastating mutation. So my main goal is to take these Alzheimer's disease risk genes that are expressed in the immune cells, or today we're going to talk about TREM2, try and link them to actual functional changes in the immune cells of the brain, so that we can then come in and develop therapies that target the actual causes of Alzheimer's disease rather than just the effect. And so to do this, I really wanted to make sure that I was modeling our human disease with human cells. And that was critically important to me because of studies like this. So this is a study that came out a couple of years ago showing the genes that change specifically in these immune cells throughout aging in humans and in mice. And it doesn't really matter exactly what these genes are, but there's a set of genes that will change as we age in our brain's immune system. And there's another set of genes in mice, but only 23 of these genes are actually the same between humans and mice. So I really didn't want to model this disease in a mouse. But since these are immune cells, it's actually critically important that we have them in their brain environment. So microglia within the brain environment are always signaling to the neurons around them and the other cell types of the brain. They're receiving signals from the peripheral immune system and the blood. And they also are responding to these neurodegenerative diseases over really long time scales. So, a neurodegenerative disease, someone's not actually going to get sick and start to lose their neurons until they're over 65 or even older. And so studying these in a dish is quite difficult because I'm not going to grow them for 80 years. But what I've done to sort of try and mix wanting to use human cells and wanting to have these cells in their proper environment is develop a chimeric system. So this starts by getting blood samples from patients. And these are actually healthy patients who don't have Alzheimer's disease. We can then take their cells in a dish and turn them into induced pluripotent stem cells. So then we have this population of cells that will divide infinitely, and we can turn them into the different types of cells in the brain or the body. Before doing that, I actually make a mutation in the TREM2 gene that I'm interested in. And so these cells here are identical, except the green ones now lack this TREM2 gene. And this is actually a model of increasing AD risk. So the people who have mutations in TREM2 that stop its function are more likely to get Alzheimer's disease. Then we can take these healthy microglia and these sick microglia or stem cells, turn them into microglia, which is a technique that I developed in my PhD, figuring out how to make stem cells into these cells of the brain. And then with another graduate student in the lab, she figured out how to actually transplant the human microglia into a mouse brain. This is a baby mouse here. If we transplant the cells before the mouse's own immune cells have come into the brain, the human cells can actually take over that empty niche and really fill up the brain. So what we end up with are human cells inside of a brain environment. So what that looks like is here. This is a cross-section of a mouse brain. On this side is where sort of the eyes and the nose would be, and this is the back of the head. We transplanted in our human microglia into the forebrain here, and you can see these green dots, every single one of them is a human cell. So there's some areas that aren't completely full of these human microglia, but we really get excellent engraftment of these cells. What we did in this experiment was actually transplant two types of cells. So we mixed together the wild type healthy microglia and these TREM2 knockout Alzheimer's disease risk microglia and put them both in the same brain just to see how they would interact with the Alzheimer's disease pathology. So these Alzheimer's amyloid beta plaques are shown here in blue, and then these are the healthy microglia and the TREM2 knockout microglia at Alzheimer's risk. And right away, looking at these images, you can see that the red cells have sort of moved and migrated around these amyloid beta plaques and seem to be surrounding them and interacting with them. Whereas the TREM2 knockout microglia are maintaining how microglia should look in a healthy brain, where they're tiled across, each having their own little region. And they don't really seem to be noticing the fact that there are these plaques and sites of damage in the brain. And so I thought that was really interesting and quantified this as well. So there actually are more of the healthy microglia are surrounding the plaques and seeming to activate in response to disease, and the TREM2 knockout cells are not. And so just to review, we're studying TREM2, one of these AD risk genes. We've now identified a function that is altered in these TREM2 knockout microglia, which is that they fail to migrate to these beta amyloid plaques. And earlier I said we'd just be able to develop therapies, but of course it's not that easy. So what I first need to do is identify the receptors that are involved and how this deficit in migration is really occurring. And so to do this more molecular work, we actually moved out of the brain and back into a dish, but still working with our human cells. And I developed this system to actually study migration in a dish. And so what this is, it's a 2-chamber system where in the center, we've plated neurons and astrocytes, other cell types of the brains that have these familial Alzheimer's disease mutations. So they're going to overproduce beta amyloid and create sort of these damage in these plaques, but outside of the brain. And then we plate the microglia, the immune cells on the outside, and ask, can they migrate in towards this damage? And so I was really excited to see that this assay in a dish was replicating what we see in the brain, where here you're just looking at the microglia in white, and the healthy wild type microglia will migrate in towards these damaged sites, but the TREM2 knockout microglia don't seem to notice that anything is happening in there that they should be responding to. So now that we had this assay, I wanted to ask, what receptors might be actually involved in this. And I ended up finding one called CXCR4, which is expressed on the microglia and responds to this cue SDF1 alpha, which is secreted by neurons and astrocytes and increases in Alzheimer's disease. And so we're seeing this protein now, this CXCR4 on the microglia in green. And you can see that the healthy cells have higher levels of this protein than the Alzheimer's disease risk cells. We also wanted to see if the functional levels of this protein were different. So if you add the stimulus, the SDF1 alpha that comes from neurons and astrocytes to the microglia, you can see they'll respond with this green. It's a calcium wave of the cell responding. So I'll play a short little video. There's where the stimulus was added, but just to pause it. Here we have more of the cells responding in the healthy condition than the disease modeling condition. And here's the quantification of that, where once we add this stimulus, these wild type cells are responding at a much higher level. So it seems like this receptor is decreased in the disease condition and decreased in its function, but we still need to link it back to this actual migration deficit. So what I did, here's the data I showed before, where we have wild type cells migrating in towards a damaged site in a dish. The TREM2 knockout cells, Alzheimer's disease risk, do not do that. But if we take the wild type cells and use a small molecule that blocks just this receptor that I think is involved in this function, we actually can see that that is sufficient to block the ability of wild type cells now to migrate into this central chamber. And so this is actually the opposite of what we would want to do in a patient brain, right? We wouldn't want to take the wild type cells and make them worse. We would want to take the knockout cells and make them better. And so the future studies would be to actually study how we can increase expression of this CXCR4 receptor in the TREM2 knockouts. And there are some ways to do this. Actually, Flonase, a flu medicine, is known to increase levels of CXCR4. So I've done some interesting study there too. But just to summarize, what we found is that wild type healthy microglia in Alzheimer's disease are using this receptor CXCR4 to migrate to sites of damage. And we found that TREM2 knockout microglia, which model this Alzheimer's disease risk, actually fail to be able to activate and don't end up moving to plaques. And so what this more broadly suggests is that actually it's going to be therapeutically beneficial to try and get more immune activation in Alzheimer's disease, since these cells where they seemed unable to respond to the disease actually increase your risk. It shows that some of this response must be good, and whether that's clearing these protein plaques in the brain or trying to compact them so they aren't touching as many of the neurons, it's not sure yet. So I'd really like to thank my lab at UC Irvine, and of course the ARCS OC. It's been really, really wonderful meeting these women and talking to them about my work. I'm actually now up doing my postdoc at UCSF, working still on these microglia, these immune cells, but a sort of interferon viral response that they can do. Thank you.
Amanda, thank you. I am just blown away with the research you women are doing, and you've touched upon so many of the things that are right in the press right now: neuromodulation and immunology and how it affects all of us. So you're describing these two wonderful things. What I would like to know from each of you, Chantel, let's say first, how many people are working on the same thing that you are right now with neuromodulation? And are you collaborating with other centers?
Yeah, thanks. Yeah, so there's a bunch of other centers that are working on this, and we're actually working with the company that makes this device also, so that they can provide us with additional data so that we can do a better job to understand why some patients are responding and others aren't. But yeah, we have collaborators all across the country and internationally as well. So there's a large team in the neuromodulation space that's trying to help this patient population and better understand how to improve this therapy.
Okay. And Amanda, what about you? How many centers are working on this, and are you collaborating?
Yeah, so definitely collaborating. It's sort of interesting. I mentioned at the beginning these new Alzheimer's disease risk genes that have all shown up in microglia. If you ask someone 5-10 years ago if they were studying the immune system in Alzheimer's disease, they would be like, why? Why are you doing that? That makes no sense. It's just responding to disease. It's not actually causative. But since these studies, there's been a huge boom in the number of people studying microglia in various different ways in Alzheimer's disease and across neurodegenerative diseases as well. And so now there's, entire conferences of people just in this field. But it's been really great working at UC Irvine, because they have one of the, I think there's 20 in the US, but they're Alzheimer's disease research centers. So we get a lot of interaction actually with patients and postmortem tissue, and being able to understand these patient-derived stem cells and things like that. So that's been a really great collaboration that I'm a part of.
Yeah. It's interesting, in our newest COVID-19, our pandemic, what we found is that the inflammatory response was what was killing people. It wasn't the virus, but the inflammatory response to the virus in the lungs. So it's amazing now that you're discovering and talking about the immunology of Alzheimer's. Chantel, back to you. And these patients who are resistant to medications, as you're doing these studies, are they off medications when you're doing the electrodes? And tell me a little bit about that.
Yeah, that's a great question. So these patients that actually identify where in the brain their seizures are starting from have undergo about a week stay in the hospital, where they have electrodes implanted in their brain that are always recording to identify where the seizures are happening. And in order to know where the seizures are happening, we have to have them have a seizure. So we take them off their medicine for that. And that's just part of standard clinical epilepsy care. But then once they get the device implanted and we know where the seizures are coming from and they get the device, they go back on their seizure drugs. So they're also on seizure drugs. And it's actually been really interesting. Those patients who have this device implanted, since it's continuously recording this electrical activity, they've actually been able to notice changes in activity when they're on certain drugs. So it's actually being used to identify drugs that might work for these drug-resistant patients better than other drugs. So there's actually been more than one use to this device because of its recording capabilities and not just its stimulation capabilities. But they are on seizure medicines as well as stimulation, yes.
Okay, thank you. Chantel, how did you get interested in the study of epilepsy?
Yeah, so I was really interested in neural engineering when I went to grad school. So I wanted to be somewhere in the field of neuromodulation, neurostimulation, because like you said, I really do think it's the future for a lot of disorders. And epilepsy is a really challenging case because some people may be familiar with Parkinson's disease and deep brain stimulation for Parkinson's disease, but you have a readout of how that patient is doing on stimulation right away. You can measure their tremor before stimulation, you turn it on, and you can measure that change. Whereas for epilepsy, seizures, we don't know when they're going to occur. And so we don't have a readout right away when we turn on stimulation if it's going to work or not. It might take up to months to notice an effect. So it's just a really challenging case of neuromodulation where we need a lot of efforts to help improve the therapy. So I was excited by this new application of deep brain stimulation in a challenging space and the potential to help a lot of people.
Absolutely, thank you. Amanda, a question for you. Who at the UCSF are you working with?
Yeah, so now I'm,
you collaborate with Penn, and have you explored the gene expression of your TRIM knockout? So your collaboration, they were asking specifically, yeah.
Yeah, so I'm now working with Dr. Martin Kampman up at UCSF on the Mission Bay campus, if you know it well. And I just started that last September, but it's been a really, really great environment as well. I haven't actually collaborated with Penn. If there's a reason why you're asking, let me know in the chat. Maybe I'll look into some new collaborations there. And as for the gene expression, yeah, so I did do, this was just sort of a little part of my project that I shared today, but a lot of gene expression studies with the TREM2 knockouts. And what we see is there's actually this special set of genes, well, this was known before my study, this special set of genes that increase with disease. They're called disease-associated microglia. And people for a long time thought that these disease-associated microglia, since they're showing up when you have disease, they must be bad, right? They're associated with disease. But what we find with the TREM2 knockout is that these cells don't turn into disease-associated microglia, sort of like you saw in the pictures. Despite the fact that these plaques are right there, they don't even notice. They stay homeostatic. And so again, what this means is that if these disease risk cells are not turning into disease-associated microglia, perhaps we actually want the disease-associated microglia to be present. So I have done a lot of gene expression studies, and they were somewhat surprising.
Well, you know, as we age, we all think we're getting it. And every time we don't remember someone's name or a date, we're like, okay, we have the big A. What's your next step in your studies? Are you going to help us, Amanda?
I'm trying my best. So a little bit what I'm working on now, so asking about gene expression sort of leads into this. So there are these disease-associated microglia that show up in the brain that we now think are helpful. And there's this other response called interferon response, which again, should be a viral response, but there's actually no virus in the brains of people with Alzheimer's disease. And so what I'm studying now is, why are we getting a viral response in this condition? And I actually think it's because as the neurons die, they're releasing some DNA, and this DNA, the cells don't know that it's human DNA versus virus DNA, so they still mount these responses. And I think actually that what's happening, and I don't have the data yet, so this is just speculation, but I think what's happening is these immune cells in the brain are thinking there's a viral response, and then they start attacking the human cells. And that's why we're getting further neurodegeneration. So I'm hoping in my postdoctoral work to prove that, and which would be very, very beneficial, because we would understand at that point that stopping this quote unquote viral response, which is not really to viruses, actually would be able to stop this brain from attacking itself.
Okay, thank you. So Chantel, what is the next step in your research?
Yeah, thanks. So my final project of my PhD work that I'm working on is we have these recordings from these patients over time. So they may have been implanted for years. And so we can, my hypothesis is that those patients who are responding to the treatment and have less seizures than they did before they were implanted, there's some electrophysiological or electrical change in their brain activity that might identify those patients who are responding. And then we might be able to use that as a tool to say, oh, we should implant these patients because we know they'll respond better than other patients. So we know it would be a good therapy. So that's what I'm looking into now, is actually looking at that recording data, the brain recording data over time, and trying to identify features of those patients who are responding to therapy.
You know, there's been so many wonderful things with neuromodulation. And so I'm hoping with Parkinson's disease and so many other things that I'm hoping you're going to be very successful with this.
Thank you.
Amanda, what do you see as your future? What are your plans?
Yeah, so my future plans are going to be to hopefully become a professor and have my own lab and keep studying neurodegeneration and these stem cell derived models of the brain. I think it's a really interesting way where we can keep true to the human genetics, but still be able to make these models in a dish. I think that's one particular challenge of neuroscience. If you're studying, cancer, for example, it's very easy to get samples of a tumor because we're taking them out anyways. But in a brain where you're losing cells particularly, you can't actually get these human samples. And I think that's been a really big deficit in our ability to study brain-related diseases, and that these stem cell-derived models can offer ways to do that.
And how long will your postdoc be? And how long have you been studying already?
All right. I'm hoping my postdoc will be about 3 or 4 years. They range anywhere from 3 years. I've met 10-year postdocs as well, but hopefully I will have good luck with the science and be able to get out before that. I have been studying neurodegeneration for quite some time. My first lab experience actually was all the way back in high school, and just sort of shadowing, but understanding the role of a different brain cell type called astrocytes in Parkinson's disease, back when we thought that transplanting these cells into the brain might be helpful for patients. And so I guess now it's been about 10 years studying neurodegeneration. It has gone by very fast, but I love it, and I'm excited to keep doing it forever.
Well, we know how long your careers take, and that's why we're so happy to support you guys in any way. Chantel, what about you? What's in your future that you would like to be doing?
Yeah, so I'm wrapping up my graduate work now. I just finished my 5th year of grad school, and I am actually doing an internship this summer in the Bay Area at Kala Health, which is a startup that has an FDA-approved device that is non-invasive stimulation of the nerves in your wrist to try to stop tremor for essential tremor patients. So I'm going to be a research scientist intern there for 3 months this summer, and then come back and defend my graduate work. So I'm really excited to get that industry experience and a new experience. And then after that, I'm going to apply to research scientist positions at a neuromodulation company. Like you said, the neuromodulation space is booming right now. There's a lot of new startups and companies who are doing research for brain stimulation for all types of disorders. So I'm really excited to contribute to that going forward.
All right, thank you. Amanda, we have a question from you, from someone who's at the Oregon National Primate Center and wondering if you have considered the monkey model.
Yeah, I actually have never worked with primates. I think certainly some of the next steps to my research would be excellent to take into those kinds of models. They're extremely useful. And again, yeah, you don't run into those problems then with the mix up between genetics, which we see between mice and humans, since the primates are so much more similar. But I haven't done it. Yeah, I'm not equipped to do it at this point. And I think a lot of the studies I'm doing are more explorative at this basic science level. And so I wouldn't want to use the number of monkeys that might be required to make these findings. But I think for next steps, it absolutely makes sense.
Okay.
Thanks.
Well, your work is amazing, impressive, and it's going to affect people in the future for years to come. So we appreciate so much. We appreciate your giving time to us today. And let me just see if we have any other questions. I don't see any more. But thank you so much for giving your time and your expertise to us today. We will have another session. Let me see. In the fall, we'll have another session. And it will start September 13th at noon Eastern time. And the latest Hall of Fame inductee, Dr. David Dixon, will be talking about research in the area of computational chemistry and its application in addressing energy and environmental problems. So we will send out a link to today's session. And we'll also send a survey, which we would appreciate your asking or answering and letting us know if there are any topics that you would be interested in hearing about. And I hope you will send back those surveys. So Amanda and Chantel, thank you again. And we will look forward to seeing everybody back in September.
Thank you all for coming. It's been really great to share research with you guys and have these discussions.
Well, you're amazing.
Thank
So
you.
Thank you.
Thank you.
Thank you. Great talks.